And in any case, the first slides usually are not so informative, so all those who will come a little bit later won't lose that much. So again, good afternoon to everybody. Thank you for being here, and let's start this talk. And as usual, we start with the acknowledgements. Usually the acknowledgements are towards the end of the presentation, but I prefer to have them at the beginning so that nobody is forgotten. And first of all, I wish to thank Mrs. Terry Aranga, her husband, and all the technical staff that make all this possible, because AutismOne is one of the most important events in the field of autism. And of course, I wish to thank you all for being here. I wish to thank my esteemed colleague and friend, Dr. Antonucci, for his contribution to the results that will be presented today. And also it's a pleasure to acknowledge the help, a special thanks to Dr. Dietrich Klingart and the staff at the Sophia Health Institute, Mrs. Daniela De Josso, for the contribution to this Ruggiero Klingart protocol tailored for autism that will be mentioned in this talk. Another special thank goes to Immuno Corporation for the development of the immunotherapy described in this talk. And also, I wish, of course, to express my gratitude for the human and scientific legacy of Dr. Jeff Bradstreet, esteemed friend and colleague, whose insights still inspire our work. And talking about Dr. Bradstreet, I am pleased to announce that in a very short while, a new book written by a South African author, the mother of an autistic child, who received tremendous benefit by applying the concepts originally elaborated by Dr. Bradstreet, will be out. There is a chapter by myself, plus many other chapters, very interesting. So I wish that you could take a look at this book as soon as it will be out. As you know, we do not give medical advice, so please do not take any of the information presented in this talk as medical advice, and please be certain to follow standard of care for each pathology, as well as rules and regulations established by health authorities in each country. I know that many of you come from all different parts of the world. Results that will be presented here have been obtained and observed in different parts of the world, so be certain not to run into troubles, and so to follow rules and regulations of the place where you live and work. I think it's also honest and transparent to disclose my conflict of interest, because I am the inventor of a number of supplements, and together with my wife, Dr. Stefania Pacini, we developed these supplements that will be described in this study. However, I must say that we take no part in treating patients, and so neither myself nor Dr. Pacini were aware at any prior knowledge of the therapies being used by the different doctors, or the details of any patient whose clinical outcomes will be commented in this study. Now, in this talk, I shall present evidence demonstrating how novel approaches to brain detoxification through the brain lymphatic system, also known as the glymphatic system, lead to significant improvements in the symptoms that are characteristics of autism. Now, my talk is divided in two parts. In the first part, I shall describe the results obtained with a novel immunotherapeutic strategy that is based on our previous clinical and experimental experience with GC-MAF, macrophage activating factor. And in the second part, I shall describe novel, unexpected results obtained with the probiotic approach that I first presented at my first participation at AutismOne in 2014. You can see that was the first slide I presented about 5 years ago. And now we have learned much, much more about this approach. Let's move, if you don't mind, to part two. Part two has been a complete surprise for us, an unexpected unknown. I'm quoting the words, famous words of former Secretary of Defense Donald Rumsfeld, who said, we know that there are things that we don't know, but then there are also unknown unknowns. Well, we knew that our probiotic approach is very helpful in autism. You can review my talks at AutismOne since 2014. You can look at the testimonials. You can speak with people who stop by at our booth and tell us the improvements they have observed with our probiotic approach. So we knew this. We also knew that our probiotic approach is instrumental in detoxifying the body and the brain. We have published papers demonstrating how the microbes bind toxins, heavy metals, and all toxicants and remove them. We have published papers. I have presented these results even 1 year ago. And since, thanks to Mrs. Aranga and her organization, all these talks are available on YouTube and the AutismOne website. You can go and review my talk of last year. So we knew all this. But we discovered that there was a big unknown. Because about a few months ago, a new tool to analyze DNA came out. It is called Axiom Microbiome Array, and it is a next-generation microarray that is a tool for the analysis of microbiome. So this was not available before. So as soon as we learned about this assay, given my personal background as a molecular biologist, we were very excited, and we asked ourselves, why don't we run an assay of this type on our probiotic, and let's see what comes out. And we saw something very, very interesting, because this assay can detect in one single shot 12,000 different strains of microbes. It's a high number, with extremely high specificity and sensitivity. And by the way, we don't run this assay. A third-party laboratory, I would say the most reputable in the world, runs this assay. And we found, to our great and pleasant surprise, that in our probiotics, there are more than 300 different microbes. So not the 5 or 6 microbes you find in a common probiotic, not even the 42 that we thought were in our probiotic and already was a huge number. No, we have more than 300. And these new results, they go and went well above and beyond our most optimistic expectations. Because using the tools of classic microbiology, that is using a microscope, we had known for years that we had scores of different microbes. So we could say, well, look, our probiotic has a huge number of microbes, 40, 42. But with this assay, we witnessed a richness in biodiversity that we could not anticipate. And this is due to the fact that the tools of classic microbiology are unable to distinguish the substrains, the plasmids and the phages, and I will talk about this later, that only the microbiome array can demonstrate. It is as if you look at animals and you say, well, there are dogs and cats, fine. But then you look in higher detail and you can say, well, among dogs you have Chihuahuas, Great Danes, German Shepherds, and a number of breeds. That's what we did. Before the microbiome assay, we could say, well, we have bifidobacteria, lactobacillus, this and that. But now we can go into detail, and just to give you an example, later on I will show you the original data. Now, as you can realize, these results put our probiotic in a league of its own, since no other probiotic can honestly make such a claim of more than 300 different species. And there is a reason for this. When we first designed this probiotic, our goal was not to make just another probiotic, but to reproduce the complexity of the healthy human microbiota. However, as we move on in this talk of mine, you will realize that it is not the sheer number of different microbes that is utterly impressive, but the biodiversity of its proprietary culture kefir grains that make our probiotic truly unique. Now, the genetic analysis revealed that in addition to the hundreds of different probiotic microbes, you know many of them, like the bifidobacteria, the lactobacilli, and so on, there is a great number of plasmids, 76, and phages, 46 phages. Now, this is, just to give you an example, of course, I cannot reproduce 400 strains, otherwise, nobody will be able to see anything. But if you take one single strain, like the Lactococcus lactis, one of the most common, you find it in many probiotics, it is as if you were saying dogs. Lactococcus lactis like dogs. Okay, good. But then you go and analyze the single substrains. I had to cut a little bit for proprietary reasons. Then you see that there is the subspecies, Lactis BV something, Cremoris CNN something, Cremoris TIFI something, a long list of boring numbers and figures that in any case identify different substrains. So there is a truly an extremely rich biodiversity. And you go down the list, you end up with, in the column 401, and you have a phages, the Lactococcus phages 712, and just a little bit above 398, you have the plasmid ATCC. ATCC stands for American Tissue Culture. 366 plasmid 1, and so many that it's impossible to mention them all. Now, what are these things? Because you know what microbes are, you know what bifidobacterium, probiotics are. You probably, unless you are a microbiologist, have never heard about plasmids and phages. Plasmids are self-replicating small DNA molecules coming from the DNA of microbes, of probiotics. And they carry genes that benefit the survival of the organism, but most important, they can be transferred to other living beings. So we can say that our probiotic not only contains a very high number of live probiotics, but thanks to the presence of plasmids, the microbes are also able to communicate with each other and with the cells of a human body. And this, of course, leads to mutual reciprocal benefits. So the plasmids, the 76 plasmids that we identified in our probiotic, they serve the purpose of cross-communication between microbes and between human cells. Thanks to the presence of plasmids, we can say that our probiotic not only has a high number of microbes, but also these microbes are not only alive, of course, but also able to talk, to communicate with each other and to our human cells. And honestly, I'm not aware of any other probiotic that can make this claim. But in my opinion, even more interesting is the presence of those 46 phages. Who are the phages? Phages can be defined as the microbes of the microbes. So we have microbes in our body, but also the microbes, they have their own microbes inside themselves. They could be called the microbiota of the microbiota. And as of today, the study of phages and how they relate to human health represents one of the frontiers of molecular medicine. For you to know, phages are the most abundant organism in the biosphere. So there are many mammals, there are many dogs, many humans, many microbes, many more phages. And as far as we are concerned on human health, they impact immunity directly in ways that are typically anti-inflammatory, and I shall elaborate on this point later. We know that phages modulate innate immunity through a mechanism that is called phagocytosis. That is how the cells of the immune system eat microbes, bacteria, cancer cells, whatever has to be destroyed. And through responses based on cytokines. And phages also impact, positively of course, adaptive immunity. So they can modulate production of antibodies and what is called effector polarization, that is, they tell to the cells of the immune system how to work, how to fight. And as I say, the impact of phages on immunity is in ways that are typically anti-inflammatory. Why do I highlight this concept? Well, Dr. Antonucci, together with Dr. Siniskalka, Schultz, and Brigida, they published just 1 year ago a paper in a very reputable journal that you can find in PubMed, describing how inflammation and neuroimmune dysregulations are associated, if not responsible for, autism spectrum disorders. So neuroinflammation and alterations of the immunity inside the brain. Now, to this end, we now know that phages have profound effects on fighting silent chronic infections. So we know that silent chronic infections are associated with a number of conditions. I would say, and I think Dr. Klingart will support my statement, that all chronic conditions, whether we're talking about cancer, metabolic diseases, of course persistent Lyme or autism, are in one way or the other associated with silent chronic infections by one microbe or the other. Well, phages, they fight these. They try to go and kill the pathogens. And thanks to the presence of phages, we can now explain the excellent results that we had observed since 2013 or maybe even earlier with our probiotic approach. Let me give you just a few examples. But before, I just wanted to highlight how phages are truly the frontiers of new immunotherapy, natural immunotherapy. This paper came out few days ago, a few hours ago, in May 2019, and here the authors, they talk about the dawn of phage therapy. So until now you probably knew about immunotherapy, you probably knew about probiotics, how the microbes help us. Well, now there is something more, the dawn of phage therapy. However, there is somebody in Russia who says, look guys, it's not so new. It is 100 years that in Russia we have been used phage preparations and we have developed them. So it could be true, could be the Iron Curtain, whatever the case. So whether phage therapy is something brand new and we are observing the dawn, or maybe this dawn has already occurred in Russia 100 years ago, we don't know. What is for certain is that it is the most advanced field of research for those who are trying to fight chronic infections, chronic conditions, as well as for those who are trying to improve the immune system and the brain immune system. Now, this paper came out 2 years ago, and it says only recently the presence of phages in the microbiome, inside our microbes, has been highlighted, and their potential role in maintaining normal immunity has gained increasing attention. So, what we are observing is something new, but is something that is already well assessed. Now, phages are the most abundant living organism on Earth. And of course, I cannot talk about all of them. So let me concentrate for the next 5-10 minutes on the number 382 of our list, 382 of the microbes in our list. It is called Lactococcus phage UL36. But then, of course, also phages are diversified just like dogs. You cannot say dogs, you have to say, well, is it a Chihuahua or a Great Dane? It makes a difference. So it is Lactococcus phage UL36 K1T1. Okay, so now we have a very good classification of what is what. So what does this microbe do? It produces a protein. It has a gene that codes for a protein. A protein that is known is called human RAD or RAD52. So the protein was first discovered in humans, and I will tell you what this protein does. And it is called RAD52. We all have this protein. And then it was discovered that this protein originated in phages billions of years ago, billions before humans came on this earth. So that's why it is called a homolog, a phage homolog. Now, we all have this protein, and this is good because this is the way the protein looks like a ring. This protein goes and looks for mistakes in DNA damage. So DNA can be damaged by a number of factors. We can be born with DNA mutations, or we can be exposed to toxicants, electromagnetic fields, ionizing radiations, all types of poisons, or maybe even spontaneous mistakes. Whatever the case, DNA has to be repaired, because if DNA breaks, you lose information and all pathologies can ensue. First and foremost, cancer. Cancer is due to mutations because DNA is not properly repaired. Now, this protein, RAD52, does exactly this, repairs DNA, looks for mistakes, looks for mutations, looks for damage to DNA inflicted by whatever, and repairs DNA. And of course, when RAD52 does not work properly, DNA is not repaired properly, And so the malfunctioning of RAD52 is associated, as it is obvious, with cancer, immune deficiencies, and now we know with autism as well. And you can find scores of papers in the peer-reviewed literature demonstrating this fact that is rather obvious. Now, we know that autism is associated with impaired or defective repair of DNA damage. You want to induce autism in a mouse model? Simply give to the pregnant mouse valproic acid, and you induce DNA damage, and the newborn mice will show typical autistic symptoms. So there is a cause-effect relationship. You induce DNA damage, you have autistic symptoms. RAD52 repairs DNA. And as you know, there is a genetic component to autism. We could discuss for hours how much is the weight of the genetic components, how much is the environmental exposure. Whatever the case, I think there is no doubt that there is also a genetic component. And RAD52 also works on the genetic component, because when RAD52 doesn't work, this malfunctioning of DNA repair is associated with autism. So it is clearly obvious that if you can supplement this protein through a natural means like this phage UL36, you have a great advantage. I think many of you know by now that autism is also associated with damage of DNA in the mitochondria. So we have DNA in the nucleus of our cells, but we also have DNA in the mitochondria, that is where energy production takes place. This is one of the many papers demonstrating how autism is associated with mutations in some DNA of the mitochondria. Well, we know, and this is another paper demonstrating this, that RAD52 is required to repair DNA in the mitochondria. Again, if you supplement RAD52 through this phage, UL36, you help repairing the DNA in the mitochondria as well. Well, I think this also is a topic somehow stolen from Dr. Klingert and others, but autism is associated with retroviral infections, HERV, human endogenous retrovirus. And again, you can find many papers in PubMed demonstrating how there is expression of these endogenous retroviruses in autism. This is a very interesting study conducted in Albania, demonstrating the expression of these human endogenous retroviruses in Albanian children with autism. So autism is associated with a number of chronic infections, and in particular with viral infections. Well, guess what? RAD52 suppresses retroviral infections of all types. Whether you're talking about one retrovirus or the other, there is one in particular I don't like to mention. Whatever the case, RAD52 suppresses retroviral infections, so it works pretty well against retroviruses. We know that autism is associated with exposure to electromagnetic fields. Again, many papers demonstrating this. Actually, this is in two parts. Autism and electromagnetic fields, plausibility of a pathophysiological link, and RAD52. RAD stands for radiation, because originally this protein was discovered because it repairs the damage to DNA inflicted by electromagnetic radiation. That's where the acronym RAD comes from. And we know that RAD52 repairs DNA damage as it is inflicted by electromagnetic fields or ionizing radiations or exposure to toxicants or everything that causes damage to DNA. This is my slide before the last, and there are many things that are born out of this observation of ours. We could conclude, looking only at one of the many phages we have found, that thanks to the presence of phage UL36 that codes for this protein called RAD52, our probiotic helps repairing DNA damages that are known to be associated with autism, either because of genetic causes or because of exposure to retrovirus, electromagnetic fields, toxicants, and everything that is in between. we could say that in our approach we exploit the driving force of evolution to help the human body heal itself. Why am I saying this? Because again, RAD52 did not appear with humans, was there billions of years before with these phages to help the microbes heal their own DNA. And we simply exploited this, and now RAD52 helps ourselves. And all these are the effects of only one of the hundreds of different microbes of our probiotic approach. And if you give me 10 more hours, I can tell you about all the others. Of course, I'm kidding. So, I thank you so much for your attention. If you want to know more, please come and see me at our booth. This is the reason why when I mention dogs, I always start with Chihuahuas, and I wait for you over there. According to my watch, we have about 15 minutes for question and answers, and I shall be more than happy to try to answer all of your questions. Thank you so much.